If you feel lonely, the advice you hear is entirely predictable. Join a club, pick up a hobby, put yourself out there. For people experiencing chronic loneliness, these well-meaning suggestions often fail because their brains have reached a biological state that makes social interaction physically difficult to process. To see this in action, we can look at scientists staffing the Neumeier 3 research station. They spend 14 months in total isolation on an Antarctic ice shelf, cut off from the rest of humanity by -50-degree temperatures and gale force winds. When researchers scanned the brains of these expeditioners before and after their mission, they found literal physical changes. The prefrontal cortex, the region behind your forehead responsible for problem solving, had lost volume. The scans also showed a sharp drop in a specific protein that nurtures the development and survival of nerve cells. Loneliness is a structural alteration. It rewires the brain in ways that actively isolate people from the very social contact they are trying to find. This need for connection is hardwired into our biology. For early humans on the savannah, Cambridge researchers tested this alarm. Volunteers fasted for 10 hours, then spent 10 hours isolated, without a phone or book. These cross-sectional brain scans show the results. The neural activation patterns from being hungry and the patterns from feeling isolated look almost exactly the same. Loneliness operates as an evolutionary imperative, a literal starvation for human contact that utilizes the same survival circuits as the need for food. When a person starves for food, their body eventually consumes its own energy stores. When a person starves for social contact, their brain finds a way to compensate. In 40,000 scans, researchers found chronically lonely people have an enlarged default network, the system activating when our minds wander. Missing real interactions. This network goes into overdrive to simulate a social life, vividly daydreaming or talking to pets. The physical architecture of the brain shifts to artificially construct the social nourishment it cannot find in reality. This survival mechanism comes at a steep cost. A socially starved brain becomes highly defensive, treating the outside world as a constant threat. Isolated people have a smaller orbitofrontal cortex, diminishing their ability to process rewards, making real-life encounters deeply uncomfortable. To protect itself, the brain's visual networks become hyper-vigilant, locking onto negative social cues in just 120 milliseconds, less than half a blink. This creates a brutal biological loop. The individual feels a painful craving for connection, but their rewired brain inherently perceives other humans as judgmental and hostile. The physical reality of the Antarctic scans and the UK biobank data reframes chronic loneliness as a structural trap. Once the prefrontal cortex has physically atrophied, escaping the cycle requires more than just willpower. Trust usually relies on a region called the insula, which processes our gut feelings. In lonely brains, this center is significantly less active, leaving individuals without the internal signals they need to determine if another person is safe or reliable. Researchers are looking towards synchrony as a way to circumvent this lack of trust. When people match their behaviors and reactions from moment to moment, it reduces the workload on the parts of the brain that scan for social threats, making interactions feel safer. Synchrony can be as simple as reciprocating a smile or mirroring body language, but it also extends to group activities like singing in a choir or rowing. By moving to the exact same rhythm, the brain begins to align with others through action rather than relying on compromised gut instincts. Transient loneliness remains a healthy hunger, a signal to seek out our tribe. But chronic loneliness requires targeted interventions, like synchronizing physical actions with others, to rebuild trust and